So let me start with the first slide. So as I mentioned you that basically I'm a physicist and I am working on a biological system. So little bit I am a biologist and currently I am working in the chemistry department. So I am applying a type of chemical principles. So many times actually whenever I am in the philosophical mood, I used to ask myself that I have jumped across many disciplines. So actually who I am. So later on actually I realized and even I feel very proud that I am an interdisciplinary person who is a physicist by mind and I apply some chemical techniques on the biological system. Okay. So that's why I will say this is actually a type of message to students. Like I came to know that most of our audience will be from the PG and the PhD students. So that's why this is a message to the PG students that if you want to do some research, you don't need any expertise or background. You just need curiosity, skill, and self-confidence. Just like me, you can see that I did my master's with physics with electronics. Then I did in biophysics. Now I am in the chemistry departments. So just like if you are curious mind and if you have a skill and if you have confidence that you can do research, so definitely don't afraid that this is my specialization in master MSc and I want to jump in different field. You are very welcome to opt any branch of physics to do your research. Just you need to be curious there. Okay. Now the thing is that like as I mentioned that we do the simulation using molecular dynamics simulation, or you can also think about the quantum computations. So the thing is that actually what we do, this story is very actually I will say that important. The thing is because as I mentioned that I do some simulations for the biological system and my basic motive that we need to know the function and mechanism of some enzymes or the proteins. So and you know that actually such type of simulations are a type of theoretical calculations. Okay. So since currently I'm in the chemistry department, honestly I'm saying that this is very hard task. The reason is that like since you people are in the physics department and you know that the thing is that like most of the physicists starting from the Einstein, Schrodinger, everyone, they were actually theoretician. So that's why for the physicist, if someone is saying that I am working the theoretical things, you will of course and by default you will get some type of respect. But the thing is that since I am working on the biological system and I am in the chemistry department, this is really very hard life because everyone like in the chemistry departments and the biologists, they used to think you are doing some theory, then they scare me from the up to down, and then say that what theory can do in the chemistry and the biology. So I will tell you with some examples. So you people know that actually our country has something like 100 nuclear bombs. This is known, we know that one. And but we also know that actually we tested just one nuclear bomb in the program. Now the thing is that just think that since we just tested one nuclear bomb in program, but we have produced 100 bombs. So now the question is that how did we make this 100 bombs while we just tested one? Why do we didn't tested 100 bombs are working or not? That is the reason. Actually, the thing is that those scientists, they actually use a theoretical model and then using the computer simulations, they tested that models in the simulations using your computational model. And when they found that this simulation or this computational model is working well, then they tested in the real world and they found that their model were correct. Now they using the same model, they can produce, they can replicate many of such models. And that's why we are confident that since the first theoretical model was correct, so that's why the other 99 bumps which we have prepared that are going to work. So this is the actually way how simulation works. Like we produce a model and then what we do, we test it in the real life and then we say that whether it is working or not. So in my research group, actually, I do similar type of things, but be aware, we are not making the nuclear bomb. We do the same things with the biological system. Just for example, we will have some type of biological models, okay? And then we using our computer simulations, we simulate all those biological models in real physiological conditions. Just for example, you know that in our body, there will be different pH conditions. And then you also know that actually in our body, there are membranes, lipids, and then there are water. So actually we used to give the same type of physiological condition and then within we test our biological models there. And many times, suppose that if I'm getting something new, then we will have some experimental collaborators, and then we ask them to test our models. So when our experimental collaborator, they found that actually our model is correct, then actually we replicate this model and we predict the mechanism of functions of desired complex. So this is how actually this computer simulation techniques, or you can see that this computational models actually works. Okay. Now the question is that, like, and you people know that from the last two slides, like I am and we are working with the computational model. Now the question is very common. Everyone knows that there are many experimental techniques. For example, you know that there are NMR, there are X-ray crystallography, and they are different like cryo-EM. So these are some experimental techniques which can give you a picture or real structure of the systems. Either it is biological system or any organic compounds. So using these techniques like X-ray crystallography, NMR, or cryo-EM, we can have the structure of those things. So that's why we have now some questions like if there are already experimental technique, why we are going to use the computational models? Okay, so let us see that why, what are the importance of this computational models? Okay, so the thing is that suppose that if you are going to take some X-ray structure of any type of molecules, so definitely that X-ray structure will give you a picture of that biological system. Just for example, I'm talking, might be during this time you people have seen in news like what is the structure of the coronavirus. You know that actually there are some spherical things, there are some spikes. So that is a type of picture taken from the cryo-EM. So these experimental techniques can give you the picture. Now I am asking you one question. As I know that most of you are from your PG and the PhD, and I also just like know that Dr. Manindra is looks like he's going to marry. So I'm asking that like since I'm assuming that theoretically you people are bachelor or the PG and PhD students, are you going to marry someone just by seeing his or her picture? So definitely not, because nowadays using this WhatsApp area and this computer technology, I am very much sure that if your mediator is showing you the picture of your could be bride or groom, definitely you are not satisfied. You will ask for some video calling, or at least you are going to meet him or her, and then you are going to decide. The same thing also happens here. Like just looking to the structure of your biological systems, you cannot see that what are the basic characteristics of that biological molecules. So actually we do the same thing. Like using the computational model, we use three-dimensional structure. We used to see their dynamics. We used to see actually their functions. Okay, so that's why I'm saying that we meet molecules using computer simulation, and we see their functions in three dimensions. Okay, so that is the basic difference, and that is actually the importance how why your computational models are better, even like if you have also experimental structures. And other thing is that you people know that like in molecules, they are never stationary. Even if you are talking about the absolute zero, still you people know that no molecules, no atoms, actually they are 100% frozen. Similarly, you also know that actually dynamics and flexibility is one of the fundamental property of life. Like in our body, you know that you used to take the water, you used to take medicines, and they used to go in our body. There are always dynamics. So that's why flexibility and dynamics is a fundamental property. So that's why you cannot explain everything just looking about the pictures. So same thing like in my research, basically I used to work on some type of enzymes, biocatalysis. So the thing is that like in these type of catalysis, you people have learned during your 10 plus 2 chemistry that in during the catalysis there are many intermediate steps, and you know that actually these intermediate steps are actually very like they are very less stable, and since they are less stable, so you cannot capture them by the experimental methods. So that's why these are the main few more things which gives you the importance of the computational models and why these computational models are actually important. Okay, next actually I will give you some example, like how these theoretical models are computational models. sometimes they give you something, they will predict some property which are far, I will say that later on experimentalists they have found. So I am giving you some examples, like this is one paper published in the JACS, and the author, like this Sason, actually he was my mentor. And you can also see Devesh Kumar. So Devesh Kumar is also alumni of our department. So actually they did some work and they published this work in 2006. Okay. In that work, actually they mentioned that there is a one mutation like selenocysteine, and it can be a very good catalyst. So later on, you can see that this work was published in 2006. In 2017, it was determined experimentally and it was published in the Nature. So you can see that actually theory predicted the same thing, something like 12 years back. Okay. And after 12 years, actually experimentalists found that things was correct. I am giving you another example. Actually, the same work like Sasan. Why I'm always citing Sasan? Because I have worked with him, so I know his work very closely. So actually, again, he predicted some property that there is one active oxidant in one enzyme, and he published this work in 2002. And you can see that same work, again, it was experimentally characterized, something like you can see 8 years later. So again, you can see that here theoretical prediction where actually uh, it was going before whatever we are getting in the experiments. Now I'm giving you the third example. The third example, actually, again, same group propose something like there are some electrostatic electric field, and this electric field can do some type of Diels-Alder equations. Now the thing is that later on in 2016, again experimentalists like Michel Coude and she published in the Nature, and they found that actually whatever they have proposed, even in her paper, she exclusively highlighted that the concept was taken by the theory, and that's why they proposed it. So these are three examples where you can think that like this computer simulations methods or computational models, they can be very useful for predicting some, I will say, some facts which we can determine later on in the experiments. So this is one type of importance of how theoretical models or how computational models actually works. Okay. So now the thing is that like there is like nowadays you know that everyone can just like in 70 to 80,000 rupees very easily one can purchase the computers, and then you can also start working with computational models. But there is also a caution, like if you are very naive in this theory or computational models, so you must be very attentive. The reason is that any theoretical model is only correct if your initial assumption is correct. Like suppose that you know that during your mathematics we start like let us assume these parameters. So assume that if your initial assumption is wrong, so definitely you are not going to get a very good results. So that's why whenever you are going to do some type of this theoretical model or computational model, your basic assumption or your initial model must be very accurate. I'm giving you one example. It is just one of my story. When I was in University of Barcelona, I was working there. So actually, I had one experimental collaborator. He was a type of bioengineer person. He used to do some modification in the genes, and then according to that modification in the genes, they can produce a different type of amino acids. So actually, what happens? They have done. They use some type of model of the computers, and then they did some theoretical calculations. Okay. Later on, they submitted that paper in the Nature Catalysis, and later on, actually, they found some reviewers had criticized that. So they contacted us, like me and my mentor, Carmi Rovira. So she is a very big scientist in of Spain. So actually, they contacted her, and then Karmi told me to again check their results. So when I used with proper force field, proper modeling, I found that whatever they have done, that is not correct. So you can read his mail that our collaborator Burns. You can see that he is mentioning that I am shocked almost by how far we could be wrong with non-optical force fields. It's mean that he was actually experimentalist, but he used some different model or incorrect model. And because of this incorrect model, actually he was getting entirely different results. Okay. But when we did that model, and when we say that using a correct model, and when then I told that actually whatever the mechanism you are predicting, that is not correct. Actually, you need to modify this amino acid glutamate. And when he tested experimentally, he was find that actually it was very fine. Like he can say that actually mutation of this glutamate was completely destroying the activity. So he was very much even shocked that how, if a wrong model, can you give me incorrect? So that's why if you are very, I will say that new in this field, so always use your theoretical model with some caution and very much sincerity. So later on, actually, I was lucky that in few time, just joining 3 to 4 months, I got the authorship in this nature catalysis. You can see just because I simulated that one. So they were very obliged even they gave me the authorship in that way. So these were some examples like how your theory can be useful or a wrong method can give you a wrong answers. Okay. So now the question is that, like, I know that actually in this in audience there are many people who are experimentalists, and there are also many people who are very good theoreticians. Just for example, Dr. Ambrees, Rastogisher, even Umesh sir. Our department, like physics department, is rich with this computational and theoretician person. So actually, you know that. So now the question is that, like, if you are going to do some type of this theoretical calculation, okay, just for example, I'm doing this molecular dynamics simulations. At the same time, we are doing some experiment. So this is well known that these experimental techniques, or whatever the experimental observation you are getting, those are actually the macroscopic examples. Just for example, if you are in using, you have some vessels, and in vessel you are measuring the pressure. So whatever the pressure you are getting, that is not microscopic property, but that is the macroscopic property of all those molecules inside your vessels. Same thing if you are going to measure the temperature. So temperature is also a macroscopic property because it is a just result of different collisions from your container and dust. So it's mean whatever you are getting in your experiments. So experiment is a type of macroscopic property. But on the same time, our simulations, like whatever we are doing in computer simulation, that is a microscopic property because we are taking a molecules of proteins, we are taking a molecules of some type of organic compounds. So the question is that we are doing the simulation at the microscopic property, but we want to get some observation which are macroscopic property. So how using this microscopic property we can correlate our microscopic observation? This might be a type of questions. Okay, so just you can see means that we want to measure the macroscopic property with microscopic simulations. So that this was one problem, or I will say that challenge. But we know that actually in statistical mechanics, if we are going to use these things, you people know that it is well proven that ensemble average in of anything that corresponds to the experimental observation. Means that if you are taking the ensemble average of all these molecular motions, that might be correlated with some type of microscopic property. So that is even a type of in statistical even I will give you example of your 10+2 physics. You people know that kinetic theory of gases. In kinetic theory of gases, when you are going to calculate the pressure exerted by on the wall of any container, you people recall that you calculate the average velocity. There will be P equals 1/3 mn v square, and that V is RMS value. So that's why even in that kinetic theory, you know that whenever you are going to take measure the pressure, it's mean you are going to take the ensemble average, means average property. So that is the in statistical mechanics. Similarly, what happens? We know that in statistical mechanics there is a hypothesis, ergodic hypothesis, and this hypothesis actually the time average of any property equals to the ensemble average. So just you can see that the time average equals to the ensemble average. Ensemble average gives you the microscopic observation. It's mean that if we are doing this molecular dynamics simulation, okay, so in molecular dynamics simulation, what we are doing, we are taking the time average, and that time average will correspond to the ensemble average, and then we are correlating with the macroscopic property. So this is how our simulation at microscopic level can give you the experimental observation. So this is basic theory about this molecular dynamic simulation. But the thing is that actually how we are going to do this simulation. So I will give a very brief introduction. So what we do, we take some initial coordinate. So luckily, initial coordinates are already deposited in, there is a data bank. You know that there are many servers, data online servers. So there is a protein data bank. Even some people who wants to do on the organic molecules, there is another database, that is Cambridge database. So you can import your structures of your molecules or whatever materials, everything from the Cambridge database. So we can import or take the initial coordinate from there. What we do, we check the missing atoms. Is there any missing atoms? Just for example, like if you are doing some X-ray crystallography, so X-ray crystallography depends upon the diffusion pattern. Hydrogen atoms are actually very small and they show a lot of the like flexibility. So that's why in your X-ray crystallography, it is very hard to capture the position of the hydrogen atom. So what happens if you are downloading any structure from this database or these servers, you will see missing hydrogens. So we check this missing hydrogen with some programs, and then we model this missing hydrogens, loops, and other any type of missing structures. What we do, we try to give them physiological conditions. For example, if I'm using a protein and I know that this protein works, this is a membrane protein, so I will soak that protein in the membrane. I will attach this protein part with the membrane, and then I will give such type of waters and ions. And then what will happen? We will assign the initial velocity, because you know that actually every dynamics that will go, they have to work with the Maxwell-Boltzmann distribution. So for that, actually, we normally assign some type of random velocity. And then what happens? We used to do some simulation. And normally, you know that actually in the real or experimental things, most of the phenomena or reactions, they used to they happens on the isothermal and isobaric conditions. So we basically give them NPT and MVT ensembles, such type of things, and then we perform some type of dynamics. Okay, so let me show you here the animation. Yeah, so you can see here, this is a one type of snapshot from my MD simulation. So you can see that after these conditions, we are doing simulations, and during this simulation, you can see that actually your proteins, even though your lipids, even your ions, they are moving. So with time, with average time, we actually extract the chemical properties or thermodynamical property, and then we correlate with the experiment. So this is protocol, like how our simulation, molecular dynamics simulation, actually it works. Okay. So the next thing, actually, I will give you just one sample. Like this is, you can see that actually this is a helix, and I have just taken this part. Okay, so you can see that actually this is organized structures. Okay, like this is a helical structure. So I just took this part, and you can see that this is not organized structure. Then I did MD simulations here. So now you can see that during these simulations, okay, sorry, this video is not working. Okay, I'm skipping this because this video is not working. Okay, so the thing is that now I'm jumping on the main topic, that how we can apply this MD simulation or computer simulation in the bioengineering. So let me give you one very example. So normally we work on the enzymes, and everyone knows that actually, like in you people know that in your high school chemistry that we have some Haber method how to prepare the ammonia. So you know that actually during this preparation of ammonia, we use iron and nickel as a catalyst. Then we apply very high temperature and high pressure. Then only you can produce the ammonia. But in our life, in our biological system, people know that our body can produce ammonia at that normal temperature and pressure, even in the natural conditions. That's why these enzymes means that most of the chemistry which we can do in at industrial level using a high temperature and high pressure using catalyst, but our body, our life can do these things in a very easy way using these normal conditions. So the thing is that people try to mimic this, or like they just try to engineer this enzyme so that they can get some products which can be of industrial and commercial use. Like using enzyme, suppose that if you want to produce something, so definitely that will be a greener approach. So that's why many of the people who are working in this bioengineering, their ultimate goal is to tweak this type of enzyme to get product which might be of commercial and industrial uses. So actually what happens using same technology in 2018, this lady, Frances Arnold, she got Nobel Prize. She is actually a bioengineer. So she used to change the gene and clone of that enzyme, and so that actually she can get the desired outcome that might be of the industrial uses. Okay. So the thing is that I'm just giving you one example. I think I'm already running out of the time. So I will very quickly go over here. So actually the thing is that like there is a some projects like we people know that in the drug things like 75% of all drugs, they actually contain some type of N containing heterocyclic rings. But the thing is that in all those natural enzymes, just for example, cytochrome P450, it does CH hydroxylation, but it doesn't do this N containing heterocyclic like CH amination. So that's why people started to mimic this or tweak this enzyme to produce such type of CH amination reactions. So actually in 2017, this Frances Arnold, she published one paper in Nature Chemistry, where actually just by doing some modification in this ligand, you can see that actually this is cysteine residue. So just by tweaking this one with serine, she found that now this enzyme, that is natural enzyme, which might have evolved from the CH oxidation, it can do the CH amination reactions. But the problem is that this type of engineering is very random. Like they have some type of setup, they will do some type of 100 mutations. In 100 mutations, they will find that one mutation is actually working. But now I'm going to give you the one example. Like if you are going to do the MD simulation, we can, before doing this engineering, you can actually predict that what is the role of some amino acids, so that there is no need to do some random mutagenesis. You can just go for a specific mutation and you can tweak the chemistry and you can get whatever the desired result you want. So just I am going to tell you that. Like this was the mechanistic puzzle. I am just skipping because of the lack of the time. Okay, this I'm giving the example. So what actually we did, I want to show you this slide. Here you can see that uh, this was wild type, means original enzyme. So C replaced this cysteine with serine, and C got one variant like P4. So you can see that its activity was very less and its selectivity was also very small. Then C mutated this alanine at 82 with lysine, and there's alanine with valine and phenylalanine with lysine. Means these are the amino acids C mutated, and then she found that actually activity increased suddenly, and selectivity was also increased by 99%. So actually what I did, we did some type of MD simulations of that wild type enzyme, and we found that during the simulation, there is one, this phenylalanine. So you can see that this phenylalanine and this substrate, you can see that actually they are actually parallel to each other. So because of this parallel orientation, actually they were applying a pi-pi interaction. And because of this pi-pi interaction, this substrate is almost captivated by this phenylalanine. So later on in the simulation, actually this phenylalanine is twisting, is rotating like from this, you can see this from the perpendicular position to parallel position. And since this substrate is captivated by this phenylalanine, so it is changing the orientation of the substrate. And that's why in the presence of this phenylalanine, we are getting this substrate very much flexible, and that's why we are getting very low activity. But when we are mutating this phenylalanine, same F2263 with lysine residue, we are actually destroying this pi-pi interaction. And because of lack of this pi-pi interactions, now this substrate is not being captivated by this any amino acids, and now it can very easily interact with this active accident, and that's why we are getting some increased activity. So now you can see that Frances Arnold did these things with random mutagenesis. Now, using your theoretical model, you can predict what was the mechanism, how actually it was working. And the good thing is that this phenylalanine and this lysine, okay, these are actually hydrophobic amino acids. They are not actually changing the electrostatic environments. So that's why they are not changing the catalysis. They are just applying some type of steric effect. And because of this steric effect, actually we are seeing such type of increased activity. So I'm just finishing that because it is already 3:40. So we also propose the mechanism of same thing using quantum mechanical calculation. I'm not going in detail of this quantum mechanical calculation because I found that actually someday Professor Rastogi, he is also going to tell you about some talk. So Dr. Ambrish, Rastogi sir, and Umesh sir, these people, even Manoj sir, they are expert in the quantum calculations. So maybe in that talk you will get some more exposure to what are these quantum mechanical calculations. So actually we did some QM calculation, and using this QM calculation we proposed actually the mechanism. We calculated the electron density, and then we found that actually whatever we have done using their experimental techniques, using some directed evolution, random mutagenesis. If you are doing such type of theoretical calculation, you can very precisely propose experimentalists that there is no need to do some random 100, 200 mutations. Just do this specific mutations and you can get a desired product. So that is basically the advantage of our theoretical calculation or these computational tools. And that's why how this theory can be used in bioengineering for a new function. Okay. At last, I would like to acknowledge DBT because I have grant from the Department of Biotechnology, and then SCL Foundation, and they are very good. Like whenever you are asking any type of money, they are always without anything they used to give you. And now currently I have these 5 PhD students, they are working, and I also acknowledge them. And at last, thank you. And if you have any questions, please, now floor is open for you. You people can ask me. Okay. Thank you. Thank you.